I'm Judy Cannon. I'm the core facility director, and I just want to start with a few slides just discussing the center, and then I'll be followed by the technical director who will get more into the details of the actual core facility. And so I wanted to introduce our center, Autophagy Inflammation Metabolism Center, which is a center of biomedical research excellence funded by the NIH. And our center is focused on the three letters of AIM, autophagy, inflammation, metabolism. Our focus is on basic research leading into translational studies, and that one of the major goals of the center is to support junior faculty, giving them mentoring, as well as the necessary equipment and technical expertise through the core facility to get them to R1 external level funding. But another major goal for the center is to develop a community of scholars focused on autophagy, inflammation, metabolism. And this is one of the reasons we developed the core centered around the types of equipment that we have, which we'll describe. And also, again, develop the infrastructure to support the community of scholars focused on these studies. And so we have the presence online. If you'd like to follow us on Twitter, go to our website, LinkedIn, and engage with us there as well. So the AIM is organized with our PI is Boyo Dretic, who introduced at the beginning, as well as our associate director, Dr. Eric Posnitz, an external advisory board, as well as an internal advisory board. And then we're comprised of a large number of faculty at the University of New Mexico, as well as, again, a team that's focused on mentoring faculty. I'll focus now on the autophagy core, which is what the scientific core. And so again, I'm the director. We have an associate director, as well as our technical director who you'll meet. And all of these activities really support the overall aims of the core and the center. So as for the actual scientific core leadership, again, I'm the director. We have an associate director, Dr. Liu, who's out of town, couldn't join us, and Dr. Larry Sklar, who was actually really instrumental in the past few years as a co-director, but now has moved to help us develop a long-term plan for the core. And then you'll meet Dr. Sharina Desai, who's our technical director, who has a lot of expertise in all our specific equipment, as well as Dr. Li Chen, who came on more recently as our associate technical director, and they have all the scientific expertise for our equipment, as well as autophagy, inflammation, metabolism. And then Dr. Casano is actually one of our mentored PIs on the AIM Center, but she's also assisting us with some of her special expertise in the core. So I wanted to talk a little bit just to let faculty and other users know about some opportunities through the scientific core. And so something that's new that's just come up, and we've really tried to be good about our communication with all the different users, is that we do have now an annual access fee, and that we ask for basically basic maintenance of all of our equipment and access to all of the various equipment that you'll hear about. And this is a flat fee annual flat fee of $400 for non-AIM members, but that we do give a discounted rate for those who are AIM members, and that we plan to gradually institute hourly charges for large equipment, but that's not begun yet. And so for those who are interested in joining us, again, one of our major goals is to develop a community here. And so we would love for anyone who's interested to become an AIM membership and get membership in AIM. And how to do this is that faculty PIs join us by basically sending a paragraph about your research interests and how they align with autophagy, inflammation, and/or metabolism, and then current CV, and then give just a short research talk to the AIM community so that it really can engage the community as to how we might be able to collaborate, interact with each other. And then that gives discounts, again, to these core charges. And so, again, we're really trying to build that community of scholars here that are focused on this autophagy, inflammation, metabolism. Also, we do have quite a few pilot awards that are available through the AIM as in general, as well as the core. And so the core specifically offers a voucher for metabolomic studies and for $5,000 towards specific studies in metabolomics. And this is done in collaboration with an outside company that we have partnered with for many years, and we facilitate this interaction. So if you're interested in doing metabolomic studies, please reach out to us and apply for this voucher, and we're happy to enable your studies to be able to do these types of studies that are done with a company. And then we also give facility use vouchers, which are funds towards supplies to use for core equipment. And so again, that's usually available annually. And so if you look for emails from our center, then these are given out for people who are interested in using equipment in core, and will help you buy the supplies that are required to use equipment in the core towards your research, as well as larger pilot projects in which PI basically can apply for a pilot project aligned with AIM that are designed to help you get great data towards external funding. So again, we have a lot of opportunities available. If you have any questions, feel free to email me or anyone, reach out to the center, and we're happy to discuss with you more about these opportunities. I will turn this over to Sharina, our technical director, to talk about the specifics.
Thank you, Judy. So we are located on the third floor of Edward Fitzgerald Hall, or FITS. Our main room is FITS 384, but we also have recently added two annex rooms, 301B, where you'll find me and I sitting a lot of times working on data analysis or helping others, and also 301A. So I don't want to take away from any of the information that we have in store for the next few days, but wanted to briefly overview our major and smaller equipment. So technically, the theme of our core facility is really centered around high content analysis. So we have two machines currently by Thermo Fisher called the Cell Insight CX7s in either a laser-based excitation platform or an LED-based excitation platform. So the LED has an on-stage incubator to allow for time-lapse microscopy, and then we also have a robotic arm that's capable of handling up to 2 stacks of plates. And basically, we have very similar but slightly different excitation capabilities. It has 7 different excitation ranges, and it can take up to a 6-channel fluorescent image. And then the highest mag that these machines can utilize is 40x. And so while you can get all the statistics from these machines and the software to complement it, we also have a very basic confocal microscope to help complement and get those higher resolution images that go with the statistics from this high content. So again, not to delve too deeply, but there are a number of bio applications that range from many, many different cellular processes where you can ask questions about cell cycle analysis or cell toxicity, micronucleus assay, invasion assays, colocalization, general intensity spot. Basically, this software can tell you the intensity, the size, the area, the location of all the pixels compared to all the pixels within the same channel, and then compare them to all the pixels in other channels. So potentially very powerful in being able to quantify your shape, your protein intensities, your mRNA intensities, whatever you can think of. And we're excited to show you all the different ways that these can be applied. So those are primarily for samples that are adherent. So adherent cells, we usually run 96-well plates. We can do slides, but we also have an imaging flow cytometer, which can also do high content analysis for samples that are in suspension. So because it's a flow cytometer in that it takes cells in suspension, but it's really a microscope. right? So all of the data is coming from images. And so we have up to 9 fluorescent channels, 2 bright field channels, a side scatter channel. So it is really the perfect blend of flow cytometry where you can get images in the thousands, but also get subcellular localization. So you can ask questions about autophagy, mitosis, others, basic cell biology questions, microbiology, and look at phagocytosis, parasitology. You can even look at cell shape changes and even the interface between 2 cells. So this is also, just as powerful. And we will be focusing on this for the next 2 days, not today. But this is our AMNIS, CellStream Mark II. We also have metabolic analyzers in a 24 well and 96 well format. So it's amazing. These, the top part of this sandwich here has a fiber optic fluorescent sensor that comes just above the cells and is able to read without fluorescent tagging, basically the glycolysis or respiration of the cells. And you can mix and match them with different drug deliveries that are delivered by pneumatic injection to be able to tell the profile, the metabolic profile of the cells. Our newest major instrument, which is the highlight for today, is the isoplexis. Formerly, we actually have the IsoLite, formerly in the company Isoplexis. And we will be highlighting, its very, very complex capability for either CodePlex or Isoplex chip analysis. And I won't say anything more since we have a special guest speaker to delve deeply into what that means for your experiment. And I just briefly wanted to go over some of the smaller equipment, not as heavy with the analysis, but still I think very helpful in supporting a lot of your work. We have an OptimaMax tabletop ultracentrifuge. We have 2 ChemiDots, which is our most highly visited machine, capable of looking at Western blots, gels, and more. We have a basic guava flow cytometer. It's just 2 lasers, but with 6 channels, it can still read a lot of 4-4 combinations. We have the nucleophector, which is really a transfection system designed to optimize primary cells are very difficult to transfect cells. And we have a variety of cell plate readers that either are standalone, so they really help with your BCA assays, your MTT assays. This citation 1 also has basic microscopy function, and we coupled that with the Seahorse metabolic analyzers in order to be able to count cells and therefore normalize your data. We also have the Emax Plus, so you can read plates all day long. And what's really nice about these plate readers is that they can read bioluminescence, fluorescence, and absorbance. We have a dual injector to be able to automate your reagent delivery. And we also have a shaping function and also temperature control. So you can even grow yeast and do a yeast growth curve assay. You can shape them in linear or orbital mode. The ChemiDocs can do very classic ECL substrate as well as fluorescent substrates. So we have 5 major fluorescent channels, which allows you to overlay, for example, if you want to look at total protein versus phosphorylated protein, and you can't resolve those sizes, that's what makes the fluorescence a really powerful tool. We don't encourage putting animals in the machine, but just to show that it is possible to look at something like GFP. And we also, of course, do stain-free normalization, which isn't as widely used, but it's a powerful technology where you can normalize not just to housekeeping genes, which, there's sometimes doubt as to whether or not your assay does not affect housekeeping genes, but you can normalize to the total protein loaded per well. We also have the OptimaMax centrifuge. So it's a tiny rotor, but it can go up to 200K times G. You can can use a density gradient, and of course, they can get things like exosomes and really anything that you need for ultracentrifugation, but it's a fixed angle rotor. So that's it. With that, I'll introduce Dr. Lee Chen, our newest addition to the core, to just finish out our introduction.
Good morning, everyone. I'm Dr. Lee Chen. I'm so proud of joining the InPort since January this year. And I'm so happy to that we have experienced lots of growth, especially in the 5 years. Here is an example of our growth from last year. Just in the first quarter of this year, we have already support over 40 labs and 80 customers, and we have reached out over 1,700 hours of usage. We have big growth, and to better support our customers, we also established our staff support booking system. We can book our time from this system, either for the group chat, we can talk about your experiments and like the sample preparation for the specific instruments, and also for the new users who wants to use our core facility, we require the core orientation to give you the preview of how to use our core and how to book our support and how to reserve the instruments. And you can also book the 1 hour training for the share equipment or the 3 hours training for the major equipment. And beside the iLab for iLab system, we also established our SharePoint web page. From this web page, you can find all the instrument information and the guide, videos, and like the instrument specs and SOPs, other tutorial things. And we are looking forward to support more customers on your research project. Thank you. And I will give it back to Sharina.
Thank you. So we've planned an exciting few days and wanted to really take the time to allow users and interested parties to take a deeper dive into some of our major instruments. So today we are going to really focus on our newest instrument, the isoplexis, and with us we have Dr. Yamilet Licon Munoz from the Picharillo Lab to talk about their work with this platform. We're also going to have later Lisa English and Keva by to demonstrate both the instrument and the software. They are held up. They're flying in from out of town, so I have yet to hear, but we do have Jamie here with us this morning. Tomorrow we're going to focus on the AMNIS, which is now a part of SciTech. And so we're going to go over some of our user data, and then also have Dr. Manuel Novin to give the tech talk. And he's also going to have open consultations and look at users analysis. So if you guys had any questions about how this machine could support your experiment, not just in a classical flow cytometry way, but to kind of delve deeper into that high content analysis. He's also going to be running a machine learning package to help users better analyze their data. Isoplexis team will also be around for further consultations, so I encourage anybody who's interested in this technology to come see them anytime in the afternoon tomorrow to talk about how they can really support your research. On Thursday, we're not going to have any major broadcasts, but we are going to be on hand to take deeper dives into the data analysis of AMNIS. Next week, we're going to focus on the Seahorse data, and we're going to be highlighting some of our major users, and I'll also get a tech talk on Zoom from people at the company. And then finally, the last day, Tuesday the 27th, we're going to be focusing on our bread and butter, the CX7. which we have lots of very experienced users, and hopefully we'll be able to show you the real breadth of different ways you can use this technology to answer many different types of questions, autophagy, information metabolism, and more, looking at various different applications. So we're gonna have their team on site and hopefully also learn a little bit about the Amira software platform that we'll be adding to the core facility. So it's a standalone 3D analytical software that works directly with the CX7 software. And also a basic overview of what our microscopy can do when we try to complement our CX7. So that's it. Most of all of this will be held in the conference room when it comes to talks, but we'll be floating around in the core facility as we highlight the machine. Thank you. Oh, I see one chat. Oh,
We go this year.
sure. We don't have it now, but we will find a way to share it in the notes as we make this available.
Do you want to just copy and paste it from, because I think of
That-
what's in the presentation.
It's in the presentation, but as it's part of the screenshot. So, yeah, we will get that on, we will be sure to put it in sometime in the future, but right now this is this is a screenshot. But it's from the UNMHSC.sharepoint.com under their different sites, and we're under UNM in core users. But yes, we'll definitely find a way to share that. Oh, wait a minute. Look at it. Thank you. Yeah, so I'm gonna stop right here and let.
No, not in the.
Thank you. I'm gonna walk away. Tell me.
Sharina, the Zoom link will be the same tomorrow?
Yes,
Yes.
thank
I'll
you.
join with the Zoom you.
Thank
tomorrow.
you so much.
Thanks.
You don't want me anywhere near you.
Oh,